Please join me in welcoming to the Distinctive Voices podium, Dr. Valentino Gantz.
Thank you. So I will start my talk, and first of all, let's get this thing to work. So I wanna clarify my affiliations. So all the work that I'm gonna be presenting today is gonna be what I've done while running my lab at UC San Diego, and as it was mentioned a second ago, I'm now a senior scientist in a small startup in San Diego, and I also have a position at Agrigene and Symbol. So I'll take you through a journey of how I went from studying evolution and development in the fruit fly to developing the concept of active genetics and then applying it to gene drives in mosquitoes and how this technology can be used to fight malaria.
Can you talk up a bit?
A little bit more? Sorry. So I'll jump in with the first, my initial interest of my journey started when I was doing my Ph.D. at UC San Diego, and I was always fascinated by the diversity in nature. And what I decided to study while there was understanding how different shapes evolve in different animals, in different animal species. And out of all this beautiful picture wing of different Drosophila, I ended up with studying something a little bit more boring, which is the one of the model organism, the fruit fly, that is an excellent genetic system to study the morphology of the wing that has this very stereotyped pattern of the wing venation. And then separately, we were trying to understand how the gene regulatory network that generate this stereotype pattern, how it has evolved through evolution in different animals that are closely related, but have a very different morphology of the wing. And so while in Drosophila, there was, as you may know, a lot of history in genetics with a lot of tools that can be readily used and accessed. For this other humpback fly, there was pretty much nothing. There was the fly and the transcriptome. And so there was very little to go along and actually generate mutations. And so that's how I ended up turning to CRISPR, which is, was at the time a upcoming gene editing system. And in its basic components, it's formed out of the Cas9 nuclease, which is a double-stranded DNA nuclease, and a guide RNA, which is a small RNA that can be programmed to have a given sequence right here that matches a given genomic target of interest. How the system acts is that the Cas9 and guide RNA come together and they find the genomic target, and then at this point, the Cas9 recognizes the site and cleaves the DNA, the double-stranded DNA, generating double-strand break. At this point, the cell cannot survive with this break in place, and so it tries to fix it through different mechanisms, and what it usually does, create a mutation at that location. And that's what we were trying to use this feature in these other fly species. And specifically, the problem that I was trying to address, I know it's a very arcane problem, was trying to understand the formation of the second vein of the wing, which is the one indicated with a green arrow. And what we know is that the determination of that vein position is done by the expression of a single gene that's called CNRPS. So when you have two good copies of gene CNRPS, everything is fine, but when you have two copies of the gene mutated, now the structure is completely gone. And the problem is that when you have only one copy, the structure is there. So it's a fully recessive mutation. And so this was the problem at the time because while in Drosophila things worked pretty well, when we went into the humpback fly, we could not identify this mutation. So we tried to find out, so I'll go back a second. we were thinking if there was a system that would allow us to transform something that was recessive, like in this case, to something that was dominant. So like something that would allow us to see the phenotype even if the system was really inefficient. And what we were hoping for is to obtain this wing, but without those two veins lacking. Unfortunately, I never got there. I got derailed in my project into a very different direction. This project was 90% there, but unfortunately remains still unpublished. So while trying to build something like this, what we came up with is with a technology that we call the mutagenic chain reaction. And this was built by bringing together the component that I just mentioned about CRISPR, which are Cas9, the guide RNA, that were organized in a very specific manner, and they were added in between two homology arms on a plasmid so that the two homology arms would match exactly the location that was targeted by the guide RNA. Now, how this system works is that the Cas9 and guide RNA come together, as mentioned previously, cut that specific location in the genome that are programmed to do so, and then the homology arms trick the cell into thinking that this is the chromosome, the other chromosome, and the cell copies whatever it's in between the two homology arms into the genome at that precise location. And this is just a delivery step. What happens then is that once this construct is inserted into the genome, now the two components are produced by the cell and can attack the opposing chromosome by the same exact mechanism. So they end up cutting it, and now the entire homology of the chromosome leads to copying of this cassette onto the other chromosome. And as you can see, this cassette becomes a self-propagating element that jumps from one chromosome to the other in a sequence-specific manner. So that was a great idea, but we had to test it. So we were working with Drosophila. We just went to our model preference, and we decided to target a very classic gene, gene yellow, which in Drosophila causes the fly to lose pigment. So a homozygous mutant for yellow, it's yellow in color, contrary to the wild type, which is darker. So it completely lacks the pigment. So when you take a classic mutant, a yellow mutant in Drosophila, you cross it to wild type, in the heterozygous animals that you get, you have full coloration because they have at least one gene that produces the pigment, and so you have a fully wild type color. We then proceeded to test whether generating a disruption of the gene yellow with the systems that I mentioned earlier would change things. And so we generated an insertional alleles of an MCR element into yellow, this MCR mutant, and we crossed it to wild type. And in this case, we were hoping that the second allele would be converted, and that is exactly what we saw. And all 100% of the flies obtained from this cross were yellow, which is exactly what we wanted to see. Now, I'm going to jump a little bit forward and like the things that we were not expecting, so we just wanted to convert something that was recessive to dominant, so from dark to yellow. But one of the things that we were, one of the fact that was unexpected in the system was the fact that this conversion not only happened in the somatic cells, but also in the germline. And every time you have a genetic element that is capable of converting the other chromosome in the germline, now you have a preferential transmission to the progeny of that genetic element. And that's pretty much the definition of what a gene drive is, which is any genetic system that is preferentially inherited. So let's go over what a gene drive is. So first, if you think about a regular Mendelian trait, let's call it this blue gene in this blue mosquito that gives it a trait, which would be similar to the yellow earlier. And this mosquito is mates with a wild type animal. Now you have that there is 50% chances of that blue gene to be given to the progeny. And by crossing these animals to other wild-type animals, you have that the frequency of this blue gene remains constant in the population following Mendel's laws of inheritance. Now, it's a little bit different when you have an element that is capable of copying itself onto the other chromosome, like we saw earlier. And in this case, if you take this green mosquito and you cross it to a wild type, you don't have 50% chances of inheriting the green gene, but you have 100% because it copied over. And so 100% of the progeny gets the green gene. And if these mosquitoes, again, mate with wild type, 100% of the progeny is green, and so on, so that you can reach almost 100% of presence of the green gene in the population. And since this dynamic changes by doubling the frequency of the green gene at each generation, this spread in a population follows an exponential growth. And we have calculated that it could go from about 1% to close to 100% in as little as 10 generations in optimal condition. And this sort of like gene spread into a population with this sort of dynamic was kind of like the holy grail that the mosquito field was looking for. And so I'll get into now on how can we engineer a gene drive that can be used to tackle malaria. But first, like, what is malaria? I want to give a little bit of a background. Malaria is a disease that's caused by a intracellular parasite that's called Plasmodium. Oh, sorry, I got too far. It's an intracellular organism that enters our red blood cells and pretty much eats them from inside, replicates, and then bursts and infects more cells. It's carried by mosquitoes and specifically targets humans. So this means that it's very specific to humans and cannot be transmitted to animals. And not only that, but it's it cannot live in the wild, so it cannot survive in water or marshes. It's an obligatory parasite, so it has a part of the life cycle within the mosquito and part of the life cycle in the person. And this gives us a very good opportunity to target this parasite, either with drugs at the level of the person or with pesticides by killing the mosquitoes that spread it. The other important concept is that mosquitoes are not born with a parasite, so it doesn't get transmitted from the mother to the eggs, but the mosquitoes need to bite someone that is currently infected to transmit the Plasmodium to another person. This disease is currently endemic in a lot of the poorest countries around the equatorial region. And while things are getting better, have been getting better over the years, there is still a lot of problems, especially in sub-Saharan Africa, where there is almost 500,000 deaths every year that usually involve kids under 5 years old. And all the improvements so far have involved a lot of different strategies, such as mechanical separation of the mosquitoes using nets, modification of the environment with pesticides that try to kill mosquitoes in the environment, as well as drugs that can be used for like curing the disease or preventing an infection. Nonetheless, even with all these different systems, the amount of cases every year have been steadily decreasing, but in the recent few years has stopped decreasing or somewhat started to slightly grow. And so what we think is that the use of a gene drive could be a completely novel tool to fight this disease. And I'm gonna now look at this green gene that we were trying to spread earlier as a thought experiment. It's something very real, and it's something that was developed here at UC Irvine by Professor Anthony James. And what he did is develop a malaria vaccine. So these mosquitoes that are transgenic for this specific gene can bite an infected person, but the Plasmodium cannot carry the parasite to another person. And so therefore they cannot be spreader. So you can imagine that if you had 100% of mosquitoes with this new characteristic, there would be no transmission, and therefore the parasite would just die off. But so now, how is this gene engineered in the specific? So once a mosquito gets a blood meal that has parasites in it, in these transgenic mosquitoes, they have to exit the stomach of the mosquitoes, but there are some antibodies expressed on the lumen of the stomach that bind this plasmodium and prevent them from exiting. And even if some of them manage to get into the hemolymph, there is a second antibody that was engineered to have a toxin that binds and kills it. And so all the plasmodiums are prevented from going all the way to the salivary glands, where then they're transmitted to the next person. And what we have done is taking this 2-gene vaccine and combined it with a gene drive. And now, there's a lot of numbers, so I want you to focus on these numbers here below, which is a summary of all the expected ratio would be 50-50 for mosquitoes carrying the transgenes and mosquitoes that didn't. But as you can see, it's way different from 50-50, but it's closer to 99% to 1, suggesting that the gene drive element is strongly converting the other chromosome and spreading within this mosquito population, in this genetic crosses, sorry. We then tested whether actually it would spread into a population, and this was done here in Irvine, and was done in a small cage population in the laboratory. But what we saw is that consistently, by adding a very small amount of these transgenic mosquitoes, about 5% of the population, in about 10 generations that would reach almost 100%, and this would be maintained, suggesting that it is possible to spread this malaria vaccine in a population, at least in the laboratory. And what we hope is that this can be also done in the wild. And now Anthony James has built at least 3 of this very efficient gene drive system, and they're evaluated to be used in the field going forward. So I've been focusing now on the gene drive aspect, which is the most obvious and most wanted tool that the mosquito field was looking for. But I want to spend a little bit on talking about what is active genetics and what we can do with it. Because when me and my former mentor saw the ability of these genetic elements to convert the other chromosome, we thought about a lot of other options that this could be used to. So we're defining here active genetics as the non-Mendelian transmission of genetic traits by means of transgenic elements able to actively convert alleles on the homologous chromosome to whatever desired state you may want. And this could be the entire transgene or a portion or even just a small sequence. And in a review that we wrote in 2015, we kind of outlined about all the possible technologies that we could try to explore, and we thought to explore at that time, the gene drives being the main one that we already had proof of concept for, but also a lot of other potential options. And so 8 years later from that initial paper, the first proof of concept in Drosophila and then the follow-up in mosquitoes, there we have published a lot of different iterations of gene drives that have all sorts of like different modification and tweaks. And here I just highlighted in different colors in green work that has been done by me or my direct colleagues, and in blue something that was done elsewhere, but still groundbreaking on their own. And then we have explored other elements. So going counterclockwise, we built these conditional gene drives that we call transcomplementing, and this is because the guide RNA and the Cas9 are not present as one transgene, but they were split into 2 different genomic locations and cannot act without the other. So only when combined, they're able to copy. And I will have an example later. So here we also have a few publications there. We've also used the copycat system, which is using just the guide RNA able to copy in presence of a Cas9 transgene. And we've used this system to go into pioneer organism, and I have a small story later talking about Culex quincofasciatus, which is another mosquitoes of interest, for which there was very little tools, very different story than Anopheles. We've also explored the use of this actively copying elements that jump from one chromosome to the other for gene therapy and cancer. And we've not gotten there yet, but we've shown that this system not only works in the germline very efficiently, but can work in somatic cells. So there you can imagine that a very efficient system to change both alleles could be used in a person to correct a gene that is broken. We've also brought this system to other model organisms. So in the fruit fly, research goes fairly fast, and it's easy to get a transgenic. But in mouse is a whole different story, and I'll show later how this can also accelerate the genetics in a model system like the mouse. We've also had a publication showing how not only we can spread a transgenic element that has the guide RNA and the Cas9, but also if the guide RNAs are designed correctly, they can be used to spread a very small gene modification. So they could be as small as a point mutation. And lastly, we have used these copycats that I mentioned also earlier as a cloning vector to study development. And actually, this publication here is perhaps the only one that came out of my PhD that's really based on the 7 years that I spent looking at development. And the very last one is instead this aspect here. We call these element erasers, which are neutralizing drives. And in this publication, we've shown how it's possible to build gene drives that can spread within a gene drive population, restoring a wild-type-like situation. And this was done as an answer to the concerns that were around the ability that we now have of engineering an entire wild population. If you can think of, if things go wrong and you have this gene drive that's going unchecked and create some problems in the wild, you may want to have in your back pocket a couple of options for stopping it if it's creating any problems. And so out of all these options that we thought back in 2015, there were also a few that kind of pop out that we really didn't think of at the time. And one was the building a drug-controlled gene drive, which is something that was actually done in my lab. And we thought about this as twofold. One, it would allow us to have more control in the laboratory, so you can basically add the drug, turn the gene drive on, remove the drug, turn the gene drive off. So it gives a lot of flexibility while doing research. But also, if you could deliver the drug in a given location, you could literally release this gene drive and have the gene drive spreading only in a given city, let's say. And so it would allow us also to control the gene drive while it spreads in the wild, and perhaps keeping it contained to a given region of interest. Another aspect that, in which my former mentor, Ethan Beer, was involved in is building some sort of analogous elements, not in fruit flies, but in bacteria. And in this paper, they show how these active genetic elements can very efficiently propagate among the multiple copies of a plasmid that are present in a bacteria. So if a bacteria is transformed with 2 different types of plasmids, the one carrying the active genetic elements slowly converts all the other ones to the same exact state. And lastly, another group did use the same very similar concept to target viruses and build a genetic element that's capable of converting other serotypes of herpes virus to the same exact condition. So if there was a co-infection with 2 viruses, slowly they got all converted to one type that carried a gene drive. And so out of all these options, I would love to talk about all of them, but it probably would take me 8 hours. So I picked 3 that kind of, I think, give a rough idea of the potential that these technologies can have. So I tried to be a little bit more diverse as possible. So the first example, it's something that came out of my lab as a very small optimization effort of this gene drive and actually turned into something a little bit more interesting and more applicable overall. This project started from this piece of data that's actually from a completely different publication from one of my star postdocs, Victor. And in this paper, what we saw is that when a gene drive fails, because like I've shown that like it can be very efficient up to 99%, but there's always a 1 to 5% in which the gene drive fails. What it does is generate small mutations at the location where it cuts. And at first, the assumption was that this mutation would be completely random, but by looking at them, which is what we did in this paper, is that we saw that these 3 specific sequences were coming up over and over and over, and the one in gray actually were pretty random. So we saw that like more than 50% of the failure event could be predicted, and this is something that later on a lot of people have observed in different systems. And so since we could predict them, we could also kind of plan ahead for it. And so what Alina did is building this transcomplementing gene drive system that I was mentioning earlier, which has the Cas9 that's split from the guide RNA at 2 different locations in the genome. And top to bottom, you can imagine that this is one individual and this is another individual. So what happens when you cross these 2 animals and these 2 elements are then combined is that you have that the Cas9 is combined with 2 guide RNAs, cuts at both locations, and usually you would have that these 2 transgenic elements copy into the other chromosome. But what happens when it fails, as I mentioned earlier, is that you have these point mutations that change the sequence and therefore prevent any further cutting. And this has been a big matter of debate in the gene drive field because this is an intrinsic mechanism of the technology that promotes resistance to the gene drive itself. But what Alina did in this specific system is adding 2 more guide RNAs that were directed to the most common sequence that we knew it was obtained after cutting. And so we have this additional 2 guide RNAs that can also be combined with Cas9. They can now cut these mutations and give the gene drive a second chance at copying itself over. And so we were hoping that this would boost efficiency. We have tested this in Drosophila, and this is exactly the cross that I just showed you. We have males that carry Cas9 and females that have guide RNAs marked with GFP. We get, in their progeny, animals that have both elements that are capable of copying onto the other chromosome. We cross them to wild-type males, and then we score the offspring. Since these have 1 copy of each, you would expect that the inheritance, if it was Mendelian, would be 50%. But instead, we see a much higher transmission to the offspring. It's 89 and 96 for the 2 locations. So one is a little bit more efficient than the other. And this is just with 1 guide RNA, as I mentioned earlier. So now when we see what happens when we add 1 guide RNA at the first location, we go from 89% to 97% up here, and you see that the red moves up. When we try the opposite and we add 1 guide RNA for the green location, the percentage goes up from 96 to 97. It's not statistically significant because it's so small. But what we did look also, if you look at these pie charts, we are measuring the amount of dots of single data points that are at 100% exactly. So you see that this goes from 38% to 48%, and that difference was instead statistically significant. When we did put all 4 guide RNAs together, exactly like the animation that I showed earlier, we see the same trend in which both values increase, both at the average inheritance as well as the percentage of data points that are 100%, suggesting that the system is working at both locations. We then did a test of whether this actually would make any difference in a gene drive setting, and seeded some of these animals in a cage, similar to what done before. And as you can see, the double tap gene drive consistently reaches higher level than the TGD control at both locations. So this, we're looking at one location here on the left and the other location on the right to compare them one to each other. So we did see that we have this consistent increase. Now, we're working with Drosophila, and that's all great, and things work. But we really thought that the concept of targeting these resistant alleles and give conversion a second chance at working could also be used in very different settings. And we paired up with the team of Alexis Komor. Specifically, this work was done by Zolt. And it's really funny because I have 2 projects in this presentation. They were both born while working out. So Alina and Zolt are my buddies from gymnastics. So we were stretching and we said, oh, we should try to do this double tap also in human cells. And so we did, Zolt did. And as you can see here, when you have the delivery of a transgene in human cells using just one guide RNA, you have a given value. When you add one secondary guide RNA, you see that this value increases. And this is comparable to what we did in Drosophila. So we just added one more guide RNA. But what Zolt could easily do in cells is adding 2 or 3 guide RNAs targeting the second most frequent and the third most frequent mutations that are created by the primary guide RNAs. And as you can see, you have a steady increase of transgenesis delivery. So this was not our, as you can see, the percentage is very low. So it wasn't the best target to look at this, but we have shown in this publication that there are other targets that give a much better increase overall. I'll now jump into the second story, which involves mice, and how the active genetic concept can be used to boost the genetics in the mouse model system. So when you have 2 mice and you want to cross them to combine 2 transgene of interest, the green one and the yellow one, what you do is you cross the mice. You have animals that are trans-heterozygous. You cross these to each other, and 1 out of 16 of their progeny should have both transgenes homozygous. You take 2 such animals, they can be genotype, cross them to each other, and you now have a new line that has both transgenes. And this is easy with 2 transgenes. It's not so easy with 3, and it gets more and more complicated till the point where, like, I think combining about 6 becomes fairly prohibitive. But what if you had that these transgenes were active and could actually convert the other chromosome? Now, in one shot, you would just cross these 2 mice, and 100% of the progeny would have both elements. And not only that, but if you cross this animal with 2 such transgenes with another one that had 4, if all these elements were active, in presence of Cas9, you would have that you cross these 2 animals, and 100% of the offspring have 6 transgenes in homozygosity. And this is the, second holy grail that we were hoping for. And we decided to test this using the mouse. And specifically, we didn't want to reinvent the wheel, so it worked really well for Drosophila, targeting a pigment gene. So we also decided to do the same here. And we targeted the tyrosinase gene, which when mutated causes the pigment of the mice to be completely gone, and therefore they're like white with a white coat and red eyes, pretty much albino. This is exactly the second project I was talking about. This project was born by a chat between me and Gunnar while we were lifting weights. So it's 2 out of 2. The project then expanded, and a lot of the mouse work was done by Hannah, an amazing grad student in the program, as well as Shannon, one of my disciples that helped with the cloning. So how the project was structured is to take these 2 mice, and instead of doing a full gene drive, we wanted to have a little bit more control. So what we generated is what's called a copycat, or a active genetic element that only has the guide RNA. So the guide RNA was inserted into the tyrosinase locus, so it was disrupting it and creating the white coat, but also had a DS red marker to mark the presence of the transgene. And separately, we didn't cross them to wild type, but we crossed them to a chinchilla mutant, which instead of having a dark color, has a more like grayish color. And this was because the chinchilla mutation was about 8 kb away from the insertion site and would allow us to distinguish the chromosome of origin to the actual copy, if that were to happen. And as I mentioned earlier, like we wanted to have control, so we had to express Cas9 at specific times, and going along with what was happening in the fruit fly, we really wanted to express the Cas9 at the time of gamete formation, so in the germline. And so we selected specific CRE lines that would drive the expression of Cas9 specifically in those tissues. So once these 2 animals were crossed, the CRE recombinase removes a stop sequence in front of Cas9, allowed to be expressed and be combined with the guide RNA, so that the guide RNA is allowed to cut at the given location in the tyrosinase locus. And so at this point, you would have that the guide RNA copies over or not. We test this by taking exactly these animals that I just described, so these ones here, and cross them to a complete wild tyrosinase null animal. And in their offspring, we had 3 options. So if the animals were chinchilla, that meant there was no cut whatsoever. If the animals were white, and there was no fluorescence, that means that the cut happened, but there was no copying. And if the animals were white and expressed the DS red, now we would have evidence of copying of the genetic element. And looking at this table, we see that this, in a lot of situations, we did have this, this combination failing and basically not cutting the wild type chromosome. In a lot of situations, we had cutting, and in few, we did have effective copying in these animals. And as you can see, it's in very specific conditions, and it's always when the Cas9 is expressed in the female germline. So we did not see in this paper any copying in the male germline. And I can't go into the details of what it was, but we did some follow-up experiments with other drivers, and we did see that it's also possible to have this conversion in the male germline to somewhat a little bit of a lesser amount. So I'll now jump to the third and very different story, which is how we can use a similar strategy to aid the conservation of specific birds in Hawaii. And this comes from a very big need on the islands of Hawaii to help with conservation, since not only birds, but all species pretty much on those islands are threatened in one way or the other by basically people being there and bringing a lot of like invasive species to the islands that are disrupting the ecosystem in all sorts of ways. But here I'll focus about what are the problems that are attacking birds in these islands. And since people arrived on these islands that have been 51 species of these birds that were extinct. And then later on, mosquitoes that target these birds were introduced by people, and 36 more species also got extinct after the mosquitoes were introduced. And right now there is 32 species that are left that are severely endangered, and only 2 that are currently not threatened. So a lot of the birds that are left don't really have a good life there, and in some cases, I think they're down to something like tens of individuals recorded in the wild. Some of these animals are beautiful, such as the honey creepers that are the most at risk currently. And the problem is that they're kind of attacked by all sorts of sides. First, there is predators that were introduced by us, such as cats, house cat, and stoats that prey on them. And since these birds have evolved with no real natural predator, they don't really know how to escape and defend themselves. Second, their eggs are targeted by mice and rats that are also invasive species, and basically kill their nest and prevent them from being from reproducing effectively. And if that wasn't enough, there's an invasive mosquito that brings an invasive parasite that these birds have never seen before the 1900s. And currently, what happens is that these mosquitoes cannot really effectively reproduce at high altitude. So there is a specific mosquito range and one where the mosquitoes do not reproduce and the birds can survive. But with global warming, this range is shrinking, and basically the birds have smaller and smaller areas at high altitudes where they can survive, to the point that conservation biologists have literally captured birds from lower islands and put them on higher islands to allow them to survive in these habitats. And the problem here is very similar to what we're looking earlier at with malaria, with the human malaria, for which there is Plasmodium, and in the case of people, it's carried by Anopheles. For birds, it's also a Plasmodium species that creates the problem, this Plasmodium relictum, and it's carried instead of Anopheles from mosquitoes with the gene Culex. And we were trying, so before I decided to leave academia, my lab was really focused on trying to address this problem, and we've taken the first 2 steps in this direction. So the first step was in a great collaboration with a few labs in Hawaii from Jolene Sutton. And we have basically started from absolute scratch, as in these mosquitoes, there was very little known. So we built all the tools, first by doing gene knockouts and identifying genomic locations where we can put a gene drive. And then second, we've developed an optimized set of CRISPR tools to efficiently edit the genome of these mosquitoes, and also created, as part of his publication, a Cas9 line that expresses Cas9 and could be used kind of like as a starting point to generate more efficient transgenesis. And the next logical step would be to generate proof of concept of a gene drive in these Culex, and then further develop an efficient gene drive for the suppression of these mosquitoes on these islands. And while the potential use of a gene drive in regions like Africa, which is a large continent, have a lot of problems, such as different pockets of subspecies that have like genetic differences, for which a gene drive could work really well in one group of animals, but not in the one in the country over. And so you may have a lot of complicated issues as a gene drive spreads in a population that's not homogeneous. While in the case of Hawaii, these islands are so small that you would have a much better chance at using a gene drive for suppression strategies. And with this, I'll conclude the talk. I'll go very quickly through my acknowledgements. First, the funding. I don't have any more of this funding, but this is all the funding that supported this work, and I'm really thankful to all of them. Especially, I want to point out the Early Independence Award. That's kind of like a R01 size grant for starting PIs, and really that helped me kick off my lab, and I really thank the NIH for that. I think it's an amazing program, and there should be a lot more support for it. I also want to thank people of my lab. They have all done amazing job while there, and also are now doing a lot of amazing work on their own. Both Shwechen and Victor are assistant professors in different locations. Alina is doing amazing things studying yeast in UC Berkeley, and Megan is going to be doing something amazing soon, as I just wrote her an amazing letter of rec a couple of weeks ago. So I'm sure she's going to do amazing. I also want to thank mainly three other labs that really were with me along the way. One, my former Ph.D. mentor, Ethan Beer, and some people in his lab that really worked on pivotal elements of bringing this technology forwards, and all the aspects that we mentioned earlier of active genetics. Also, Anthony James, collaborator from UCI, and people in his lab that really focused on building the best gene drive possible for Anopheles, and they really did amazing work, as well as the Comor lab, with whom I did that small transition from Drosophila to human cells, and with whom we had an amazing collaboration. I also want to thank all the members of their labs, not just them. I just wanted to point out the key people that were involved in the work that I presented today. And not only their labs, but at UCSD we built a much larger community around this technology, and we really had groups that were working from Drosophila mosquitoes to potentially use this for human therapy, mouse you've seen, as well as fish, plants, and yeast. So it was a huge community, and I'm sure I'm missing someone here as people came and went over the years. So with that, I'll take questions.